Hi, everyone. We are now on chapter 9, talking about the cell cycle and mitosis. This video will also feature what happens if mitosis goes wrong and the cell cycle goes wrong, and we'll end the video talking a little bit about cancer. So this video will cover all of these you must knows. All right, so let's talk about cell division. Once upon a time, you are one single cell, and today you are anywhere from 36 to 37 trillion cells. So how did we get here? First, we are going to be learning a lot of new vocab and concepts in this chapter, but here's some enduring understandings, things that you really need to understand in order for everything to connect. So the first is cells communicate by generating, creating, transmitting, sending, and then receiving and responding to chemical signals. So one cell will either create or generate a hormone or a neurotransmitter, and it will send it out, and it will be received by another cell, which will respond. It'll maybe make that cell go through apoptosis, cell death, or it will turn on some enzymes, or it'll tell the cell to start dividing. And that's how cells are communicating with each other. The second, timing and coordination of biological mechanisms, things happening in cells, involved in growth, reproduction, and homeostasis depend on organisms responding to environmental cues. So here's the key thing: the environment around you impacts you as a living thing. How? Well, it tells your body, hey, when is it time to start growing? For some species, hey, when is it time to start reproducing in your lifespan? Or when is it time, or how should we coordinate homeostasis? How should we take our temperature and bring it back to our set point if it's too high or if it's too low? So this is all connected to stuff that's happening outside of us. And lastly, heritable information provides for continuity of life. So for life to continue on Earth, we need to be able to pass information from one species or one organism to the next organism, so that life can continue on planet Earth and doesn't stop when an organism dies. So key things, we're going to add on some vocab and some details as we go throughout the video. So firstly, cell division. What is it? Why do cells need to do it? Well, firstly, for some organisms, like unicellular organisms, this is how you make new offspring. So for example, an amoeba. Amoeba here is going through asexual reproduction. It's creating a copy of itself. It's a single-celled eukaryote, and it's dividing into new cells. Each of these new cells will now be its own individual organism. The second reason you might need to do cell division is for growth and development. So you started off as one cell, and now you're 36, 37 trillion. How do we get there? Well, the first cell is going to actually divide. So right after sperm and egg combine, that cell will divide, and then those cells will divide, and those cells will divide until you eventually get a multicellular organism. And lastly, tissue renewal, or repairing or creating new cells. So for example here, we have bone marrow cells and they're dividing. And when those cells divide, they will create new red blood cells. Now, a lot of the cells in your body are able to divide and create new cells, except there are a few that don't. So for example, your brain cells or your nerve cells, they can't make new cells. They can't make daughter cells. Also, your muscle cells, you can't make new muscle cells. Muscle cells can only grow or basically shrink, get smaller, but you can't grow new muscle cells or new nerve cells. All right, so here let's take a look at a picture of a cell under a microscope. You see in the middle, we have these blue pieces that have been stained. These are chromosomes. And on the outside, we have this red piece right here. This is the cytoskeleton of the cell. And notice how it's kind of impacting the chromosomes in some way. This cell is a lily cell from a flower, and it's getting ready to divide. So in this video, we're going to zoom in on what's going on in this process. How can the cell divide to make new cells? Here is another picture just so you can see. This is a rat kangaroo cell. And notice here in the blue, we have these things called chromosomes. And the chromosomes are attaching to these red pieces and these green pieces here, the cellular machinery, these proteins here and these proteins here. And something's going to happen to these chromosomes. This cell is going to pull these chromosomes apart. And this is a picture of it happening under a microscope. So let's talk a little bit about what DNA is. When we're talking about chromosomes, we're talking about DNA. And let's zoom in really, really, really closely into a chromosome. If you were to zoom in really closely, here's what you would see. You would see a long string, and this long string is DNA, deoxyribonucleic acid, right? And so this is a long string in eukaryotic cells. In prokaryotic cells, DNA is in a circle. Now, this DNA, if it was just in your cells, all jumbled up, it would get really, it would get tangled, and it would basically not be very organized. Also, in your cell, in one of your cells, you have about 2 meters of DNA. So if you took all your DNA in one cell and you stretched it out, it'd be 2 meters long. Now, your cell, that's 250,000 times your cell's diameter. So how can all that DNA fit into one cell? Here's how. It's going to condense into something called a chromosome. Condense means to kind of coil up and make it even smaller so that it's one chromosome. Kind of think of it as like string in a yarn. So if you took string and you unwound it, you could stretch it out really, really far. But if you wind it up right, you can create a ball of yarn that's a lot more condensed or smaller. So let's kind of take a look at how this works. So you have a long string of DNA, and it's going to get wrapped around these proteins here called histones. Now, if you have DNA plus protein plus histone, we call that chromatin. Chromatin. Now, if you have multiple histones together, this is called a nucleosome. Okay, don't worry about memorizing that here. If you condense that even more, you're going to create an entire chromosome. So all the nucleosomes will be wrapped around each other until you make the whole thing the chromosome here. So chromosome is protein and DNA all wound up together, tightly packed and condensed. So here's a picture under microscope. We call these pieces right here, we'll come back to this more. This is called a chromatid. So it sounds very similar to chromatin. Chromatin is protein plus DNA. Chromatid is a piece of a chromosome here. And notice we have these two, they're called sister chromatids. They're very similar, they have the same DNA structure. So if you were to unravel this top chromatid and this bottom chromatid, they would have the exact same DNA. And they're connected at the center by something called a centromere. And each centromere, or the centromere connects the two sister chromatids. Now, in eukaryotic cells, different species have a different number of chromosomes in the nucleus where the DNA is held. So, but there are some differences in the different types of cells that you have in your body. So for example, let's look at a human, and then we'll look at some other examples. Most of your cells in your body are non-reproductive cells or somatic cells. These are your body cells, like your skin cells, your muscle cells, your blood cells, your bone cells, your white blood cells. Most of the cells in your body, they're somatic cells. And you want to write down this word. This is an important word. Now, in every single one of your cells, in your somatic cells, inside the nucleus, if you were to go inside or take a picture under a microscope, you would see 46 chromosomes. Now, the chromosomes are going to be paired up, and we'll look more closely at this later, but they're going to be paired up together because for each pair, you got one chromosome from your mom and one chromosome from your dad. So if you had 23 pairs, right, each one came from mom and one came from dad, then you would have 46 total chromosomes. However, in some of your cells, called your gametes, these are your reproductive cells, so either sperm cells or egg cells. You can't have two sets of chromosomes. You can't have 46 chromosomes, right? Because think about it. If your sperm cell had 46 chromosomes and it combined with an egg cell, that would be way too many chromosomes for that new cell. You should only have 46 chromosomes in one of your cells. So these reproductive cells have half as many chromosomes, 23. Okay, so we call this the haploid number. It's the number of chromosomes in a gamete. We call this the diploid number, the number of chromosomes, di means two, in your somatic cells. Okay, so in humans, we have 23 chromosomes in our sperm and our eggs and our gametes, so that when the sperm, 23 chromosomes, plus the egg come together for fertilization, we have 46 chromosomes in our new cell, in our fertilized egg. All right, now some species have different amounts of chromosomes. So for example, cabbage, right? Cabbage is a plant, it's a living thing, has chromosomes inside the cells, has 18 chromosomes. Chimpanzees have 48 chromosomes, so very, very close to humans. Elephants have 56 chromosomes. Hedgehogs have 90. So I used to think that organisms that were maybe more complex or more intelligent had more chromosomes, but that's not true. There are species of algae that have 148 chromosomes in every single cell. So that's the case for different species. All right, so let's take a look at a practice question here. Let's say you were counting the number of chromosomes in the gametes and the somatic cells of a giraffe. Okay, so we don't know how many cells or how many chromosomes a giraffe have, but let's think about the ratio. What should be the ratio of the number in the former, so in the gametes, compared to the latter in the somatic cells? So think about it. Is there going to be an equal number, 1-to-1, of gametes and somatic cells? Is there going to be half in gametes and double in the somatic cells? Or C, D, or E? Okay, what's the ratio? Well, take a moment, pause the video if you need to, think about what the answer might be. If you said B, 1 to 2, you're correct. So we have half the number of gametes, of chromosomes in our gametes compared to our somatic cells have double, right? Our cells have 23 chromosomes in our gametes and 46 chromosomes in our somatic cells. So we have double the number of chromosomes in our somatic cells. All right, let's do another practice question. We'll look closely at this, but I want you to take a guess for now. How many chromosomes are in the middle cell? So remember, the whole point of cell division is to create new baby cells, but we need to make sure that the right amount of DNA gets put in each one. So let's look at this top one here. This is one chromosome, right? This is one chromatid, one piece, and it has one string of DNA. Now, when it's time for the cell to divide, it's going to create a copy of this DNA. So now you have two strands of DNA. But remember, each of these strands is condensed into a chromatid. So you have two sister chromatids. They're exact same. This DNA has just made a copy of itself. Now they're called sister chromatids. How many chromosomes are in this cell? Is this still one chromosome? Is it two chromosomes? Make a guess. And then these sister chromatids will be divided when the cell divides into two separate cells. So now our little baby cells, our new daughter cells, both have the DNA that originally came from the parent. And this DNA is exactly the same. So take your guess. 1, 2, 3, or 4 chromosomes in the middle cell. If you said 1, you're correct. And this is where it can get a little tricky. Even though this looks a little bit different, it's still a chromosome because it has the exact same DNA on this side, and they're connected together at the centromere at the center. So this is still one chromosome. The only difference is now the DNA has been replicated or duplicated here. Now we have two strands of DNA that are all condensed instead of one. Okay, so to look closely at that, we already kind of talked about it, but one chromosome, we call one piece a chromosome arm. Don't worry about memorizing that. Here we have the centromere holding the sister chromatids together. During DNA replication, this DNA will be copied, it'll be replicated. So now you have two copies, and the DNA will be condensed. So now you have two condensed copies of the same piece of DNA, the exact same piece of DNA. We call this a sister chromatid, and this is still just a chromosome, okay? And then now we separate them. We separate the sister chromatids and put each one in one cell, and now they both have the same exact piece of DNA. So how does a cell know when it's time to divide? Well, every cell operates on its own schedule, and this schedule is called the cell cycle. And I like to compare the cell cycle to the seasons in a calendar year. So what we see here is a model of the cell cycle, and let's compare it to a calendar. So each part of the cell cycle is called a different phase. So we have G1, S, and G2. Each of these three combined makes interphase. And then we have the mitotic phase, which is split up in two pieces called cytokinesis and mitosis. So think of the G1 phase as kind of like winter at the beginning of the year. Then we have the S phase, that's spring, and then summer, and then fall. Winter, spring, summer, fall. And it keeps going and going and going. Just like every year, we have the same seasons. And in each phase, something different is happening inside the cell. So let's zoom in first on interphase. Interphase is made up of three smaller phases, G1, S, and G2. Now, if you were to pick any cell from your body and look inside, most of the cells are in interphase because 90% of the cell's lifespan is spent in interphase, and then only at the very end, a very small portion, 10%, is the mitotic phase, and this is actually where the cell is dividing. So let's focus in on interphase and look at the first part, G1. It's also called GAP1. Scientists used to think that in the GAP phase, nothing was really happening in the cell, but now we know that things actually are happening. So in the G1 phase, we have metabolic activity and growth. So the cell is metabolizing, it's taking in food, it's taking in glucose, and doing cellular respiration to make ATP. The cell is growing. Also, different cells in your body have different functions. So maybe some cells are creating proteins that are going to be hormones that are going to be secreted. Maybe your white blood cells are working on destroying invading cells. Maybe your muscle cells, they're expanding and contracting. So different cells are doing different things, but this is kind of like the normal time in a cell. Everything's kind of functioning as normal. And if you were to go inside the cell and look inside the nucleus, you would see for a human, a somatic cell, 46 chromosomes, they're unduplicated. Then we enter into the S phase. This is called the synthesis phase. The cell is still metabolizing, it's still growing, but now we're going to make a copy of our DNA. Now we have duplicated chromosomes. So originally we had 46 singular chromatids. Now they're all going to make a copy. Now we have 46, but now they have sister chromatids. They look a little bit different. They're duplicated. Then we get into our G2 phase. We have metabolic activity, it's still metabolizing. It's growing, but now the cell starts preparing for cell division. So GAP2 phase. Then we get into the mitotic phase. This is where the cell is actually going to divide. And there's two phases here. First is called mitosis. Mitosis is where you take those two sister chromatids and you're going to split them apart. And each of those chromatids will go into the new daughter cells and create two new nuclei. So you're going to have, basically, if you have your DNA, it's going to be separated into two sides of the cell. And you're going to have one nucleus on this side and one nucleus on this side. And eventually that will create two new cells with their own nuclei, with their own set of DNA. So mitosis is just the distribution of the chromosomes, so it's basically splitting the DNA. And then cytokinesis is actually dividing the cytoplasm. So you have a cell with two nuclei, and then eventually, boop, the cytoplasm splits, and now you have two daughter cells. And now those daughter cells can now start their own cell cycle. So you split the cells, and then your next daughter cell, it will now go into G1. It'll do its normal activities. It will grow. It has 46 chromosomes. Then it will make a copy of its DNA. It will do DNA synthesis, S phase, and then it will prepare for cell division. And then that daughter cell will divide, and then that cell will go through cell division, and that cell will go through cell cycle, right? And it continues and continues, and that's how we get more cells. Okay, now let's focus in just on the mitotic phase, and let's look closely at mitosis. We can actually split mitosis now up into 5 steps. And these steps you need to know the names of, and you need to know what happens in each step. So we call it PMAT, or prophase, prometaphase, metaphase, anaphase, and telophase. And then finally, cytokinesis is splitting the cytoplasm. Let's take a look at each of these phases closely. I'm going to zoom in here a little bit. So remember, at the end of interphase, we ended with G2. And G2, the cell is starting to prepare to divide. And let's look closely at what's going on. This is a simplified cell. We've gotten rid of all the other organelles, and we're just focusing in here on the nucleus. On the outside, we have our nuclear envelope. We have on the inside the nucleolus. This is the part of the cell or of the nucleus that creates ribosomes. Then we have, here's our genetic material. We have our chromosomes. In your cells, you have 46 in your somatic cells, but now they've been duplicated. So now they all have sister chromatids, and they're uncondensed. So they're actually all just strings. They're not all condensed yet. You also have these important players called the centrosomes, and these are going to be helping to split apart the chromosomes eventually. And these pieces here are centriole pairs. So 2 centrioles make a centrosome, and we have 2 of them. All right, now, once we're done with G2, we've prepared for mitosis. What we're going to do, and how have we prepared? Well, basically, we've made a copy of the DNA, right? So now each chromatid now has 2 copies of the chromatid, sister chromatids. We're ready to divide. All right, so prophase, the first step of mitosis. Now we're in the M phase of the cell cycle. What happens here is our chromosomes here are going to start to condense. So now we have 2 sister chromatids of 1 chromosome. So here's 1, 2, 3, 4, 5, 6. There's, right, in your cells, you have 46 of these, but in this example, we're just simplifying it. So your cells, prophase, the chromosomes condense. That's what you need to know. And then your centrosomes here, they're going to start to create something called spindle fibers. Spindle fibers are long chains of proteins or fibers that are going to act kind of like fingers eventually to pull apart the DNA. So that's the important part. You don't need to know this. Okay, prometaphase. Pro means before, so it's like right before metaphase. What happens? Well, first, here we had our nuclear envelope. Notice that it has dissolved. So it has actually disappeared. We're getting rid of our nucleus, and it's being dissolved. We still have our condensed chromosomes. Now, our spindle fibers are now going to be latching on here to our sister chromatids at a very specific place. So see here this vocabulary word, kinetochore. So a kinetochore is a protein that's on the in the center of the chromosome here that the spindle fiber is going to connect onto. It's going to hook on. Then we have some of these kinetochores that are actually not hooking onto chromosomes, and their job is to basically elongate the cell, make it longer. So prometaphase, kinetochores connect to spindle fibers, the nuclear envelope dissolves, and we're ready to go to metaphase. Okay, metaphase, this is my favorite part of the cell cycle. I just think it's kind of cool. We have our chromosomes are going to line up. They're all going to get in a line at the center of the cell. And the center of the cell, we call it the metaphase plate. It's kind of like a plate. It's where they're all kind of spread out in a line. Now, it's not actually like sitting on a plate, but we call it the metaphase plate because they're all aligned here. Here's our centrosomes at each pole. We call them poles because they're opposite sides, kind of like the North Pole and the South Pole. And now remember the spindle fibers. There's a spindle fiber. It's connected onto the kinetochore, and it's now latched on. It's ready to start pulling the sister chromatids apart. So metaphase, chromosomes light up at the metaphase plate, and centrosomes get to the poles, and they're ready to start pulling apart the chromatids. Okay, anaphase. Now, at anaphase, we're now separating the sister chromatids, and we do that by taking these spindle fibers, and the spindle fibers are going to shorten. So the spindle fibers are made up of a long chain of proteins. That chain is going to shorten and shorten and shorten, and it's going to pull the chromosomes to each pole of the cell. So anaphase, the chromosomes are splitting. And now you have half the chromosomes on one side of the cell, half on the other side. So each daughter cell will get an exact copy. Okay, next phase is telophase. So telophase here, we have our nucleus is actually reforming. So our nuclear envelope. So we're going to now create two nuclei. We have this thing called a cleavage furrow. So notice that the cytoplasm is starting to pinch off to make two separate cells. And notice that our spindle fibers, these guys here, have been detached. And we have our centromeres. They're just kind of hanging out here. We have our nucleolus. This is where you make your ribosomes reforming. Okay, and then cytokinesis, the last step, we don't see it here, but the cytoplasm will officially split in half, and now you have two daughter cells, and each daughter cell has the exact same DNA as the original parent cell. Why? Well, because in the S phase of the cell cycle, that original cell just made a copy, and each copy is now in daughter cells. So how does a cell know which phase of the cell cycle it's in? Well, to understand this, scientists did an experiment where they took two cells and they were both in different phases of the cell cycle. This one in the S phase and this one in the G1 phase, experiment 1. And they fused the two cells together. And when they fused the two cells together, the cell that was in the G1 phase actually moved into the synthesis phase and started synthesizing DNA. Then they did it again. They took a cell in the M phase, so it was doing mitosis here. And right here we see prometaphase is happening right here. The spindle fibers are forming and the chromosomes are condensing. And then they took a cell in G1 and they fused them together. And they noticed that the G1 nucleus immediately began to do mitosis. So we see spindle fibers forming, the chromosomes are condensing, even though the chromosomes haven't even been duplicated yet, right? G1 comes before the S phase. So these chromosomes here haven't even been duplicated. So what is it that's causing the cells in G1 to move into the S phase in this case or the M phase in this case? What are these factors? And scientists predict, well, maybe there's some sort of molecular signals. Maybe there's some sort of molecule that's telling the cell and communicating to the cell, hey, it's time to move into the next phase. So let's explore that here. So scientists add these pieces to our cell cycle model. And remember, what this is showing is basically like a calendar. So even though it's in a circle, this is just showing the passage of time for a cell, and it's in a cycle, so that's why they put in a circle. But we see here that after the G1 phase, so this is like normal cell life, it's doing metabolism, it's growing. At the G1 checkpoint, there's going to be kind of like a red light, green light, if the cell can move on into the rest of G1 and then start moving into the S phase. So there's a checkpoint here. Now, what does this mean? It means something's going to happen inside the cell in order for it to actually move into and to start synthesizing and making a copy of the DNA. And then notice here at the end, so again, we're looking at time, so time keeps going. We get into the S phase. What's happening in the cell is the cell's making a copy of its DNA. And then we get into the G2 phase, so we're continuing to grow, we're metabolizing, we're preparing for mitosis. And then right here at the end of G2, we have our G2 checkpoint, where if there's a green light, then we can move into the M phase. Or mitosis. If there is a red light, then the cell will stop. And we'll learn why this is important in a second. And then even here, there's a checkpoint within mitosis called the M checkpoint. And what does this mean? What is controlling the cell? What is telling it it's time to go and when it's time to stop? We'll continue to explore that. Okay, so we call this a cell cycle control system. Like I said, our cell cycle control system is regulated by internal control. So there's something happening inside. There's some kind of molecule inside that's telling the cell what to do. And then even external control. So there could be something outside of the cell that's communicating. Remember, this is one of our enduring understandings. Cells respond to things in their external environment, and those are signals telling the cell it's time to do something. So external or internal signals. And these checkpoints tell the cell when it's time to go or when it's time to stop. All right, now for many cells, the G1 checkpoint is the most important. So if the cell gets a G1 checkpoint and everything's good, it's a green light, the cell will move into the S, then G2, and the M phases, and the cell will divide. Now, if the cell does not get a green light for the G1 checkpoint, it will move into something called the G0 or the G0. What this is, it's basically like a resting state. The cell will never move along into the S phase or the G2 phase. So if we look, we'll take a look here. So if there's a red light and there's a certain type of red light, then the cell will stop. It will go into G0 and it won't move into S and G2 or M. And what kind of cells do this? Well, for example, your brain cells. So your brain cells stay in G0. They're never going to synthesize DNA. They're never going to go into G2. They're never going to do mitosis because brain cells, they don't divide. Same thing for muscle cells. They're not going to divide and make new cells. So be thankful for the brain cells you have. Try not to destroy them because you're not going to get any more. So G0, again, is if the cell will not divide. It will just kind of hang out in its normal state until apoptosis, which is when the cell is going to basically kill itself and die. All right, so let's look at these checkpoints a little bit closely. The first one is called the G0 checkpoint. And here we're looking at the cell cycle in the G1. So once time has passed and we get to this point here, there's going to be some sort of signal. And it's going to tell the cell, if it's a no, it's going to tell the cell, hey, time to go into G0. Let's not divide. Let's just kind of chill for the rest of our life. But if the cell does receive a green, a go-ahead signal, then it will continue in the cell cycle. So that's the G1 checkpoint, and this is an important checkpoint. So cells, if they don't get the go-ahead, they just chill for the rest of their life. Their fate is not to continue to divide. Most cells do, though, continue on. And then we have our second important checkpoint, which is the M checkpoint. And let's look at this one here. So right before metaphase, we have prometaphase. And what happens is the cell in mitosis will get a stop signal if any of the chromosomes are not attached to spindle fibers. So remember the kinetochores, those proteins in the chromosome that attaches to spindle fibers, and then those spindle fibers pull the chromosomes, it's the individual chromatid, each chromatid to each side of the cell, each pole. If some of the chromosomes aren't attached, and the cell cycle stops, which is kind of cool, right? Your cells know, hey, we don't have everyone on board. We got to wait till everyone's on board. So how can we get past this checkpoint? Well, the cell will wait. And once all the chromosomes are attached to spindle fibers on both plates, so on the plate, each sister chromatid is attached, then the go-ahead signal will allow the cell to move into anaphase. And so the sister chromatids will start being pulled to each side of the cell. So those are the two checkpoints here. Now, another thing that's going to help us understand more, like what are these signals, what's happening inside the cell, is this experiment that was done. So scientists took a group of some tissue, some connective tissue. This is a group of cells, and they cut it up in a petri dish. And then they used some enzymes. Basically, they broke apart all the stuff around the cells, the extracellular matrix, so everything around the cells. So now you just have like a suspension of cells just kind of like separate from each other. Then they took these cells, this culture it's called, and they transferred it to this culture vessel, and it contained a basic growth medium. Basically, it's like sugar, some glucose, amino acids, some salt, so things that keep the cell alive, so make sure it doesn't die, and some antibiotics, so there's no bacteria growing here. And they just kind of let it sit. So this is like our control. Then they did another experiment where they did the same thing, but they added something here called a platelet-derived growth factor. Now, this is a molecule that they added to the vessel, and then they waited. They let it sit, they incubated it, so they put it at a certain temperature for 24 hours. Now, without platelet-derived growth factor, without the molecule added, what happens? Well, without it, the cells didn't divide, they just hung out. But when you added this molecule, what happens is the cells proliferate. That means they grow, they make more copies. And what we see here is with the growth factor, now we have more copies. So what does that tell us about what is causing the cell cycle to go through mitosis? Well, maybe there's some molecule that tells a cell, hey, it's time to turn on or turn off one of the phases of the cell cycle. All right, here's another experiment that was done that kind of helps us understand, like, what is it that's causing the cell to know when it's time to divide? So in this experiment, scientists took cells that are anchored to a dish surface. So we had a dish here, and the cells here are kind of suspended on their own. And they noticed that they let the cells sit for a bit, and they noticed that the cells would form a complete layer. So they formed one layer at the bottom of the dish, but then they stopped dividing. So this is called density-dependent inhibition, which means once the cells are all touching each other, once they notice that every space has been filled, they're going to notify each other that, hey, it's time to stop dividing. So number one, they're communicating with each other. And number two, they're controlling their cell cycle. They're saying, hey, it's time to stop dividing. Let's stay in G1 or go into G0, or just wait for a little bit so that we don't divide more. We're taking up enough space. And think of why that's important, right? Your organs, they don't need to always generate new cells. Like if your heart was constantly making copies of itself, or your muscle cells don't make copies anyway, but if your certain organ was making too many copies, then you would have too much, right? You don't need that much tissue. You just need enough. So this is called density-dependent inhibition. But they scraped away some cells and they saw, okay, what would happen? Do the cells think that there's still enough cells covering the bottom? And no, they kept dividing. So they're able to signal to each other, hey, it's time to start dividing. We need to fill this space. Okay, so that's what's happening here. Now, this is just with normal cells. So these are cells of a mammal. So the cell's density is limited to a single layer because they're contacting each other, and they all have enough nutrients and they're able to survive. But in different cells, cancer cells, they tried this again, but what they found that the cancer cells actually continue to divide beyond the single layer, and they form a clump of overlapping cells. So they don't exhibit anchorage dependent. This anchorage dependent means they need a surface. They need a surface to bind to, or density dependent inhibition. So even though basically all the cells have taken up the space, it's dense, right? They're not depending on that density to tell them to, hey, it's time to stop dividing. So they'll continue to divide and divide without stopping. So something has gone wrong. The cell cycle is not being regulated. Okay, so we just talked about this. Here it is in words if you want to pause and look a little bit more closely on it, but basically what I just explained. All right, so just to review, these checkpoints G1, G2, and M tell the cell, hey, it's time to move on or time to stop, freeze. So everything is timed, right? And this is part of our enduring understanding too. Everything is timed. Certain times it's time to do certain things, but what is it that's doing that? There are tiny molecules in the cells that are telling the cell, hey, it's time to move on or not. And we'll look closely at that in the next piece here. So what is it at each of the checkpoints that tells a cell green light or red light? We're going to call them for now go-ahead signals, and these are signals that promote cell growth and division. Well, what is a signal? We learned in the last chapter that a signal is a molecule that will be received by a protein, and then it will be transduced. The signal will be transduced or carried, and then some response will happen. And the response will be, okay, move into the next phase of the cell cycle. And there are two types of these signals, intracellular and extracellular. An intracellular signal is also called a promoting factor. And what happens is a molecule can be activated, and this activation of the molecule can signal to the cell, hey, it's time to start moving into the next phase. That is the response for signal transduction response for intracellular signals. Or you could have an extracellular signal, and this would be where you have another cell in the body that is secreting a hormone that travels to the target cell, acting as a ligand, right? And it binds to the receptor protein in the cell membrane. And that receptor protein will then transduce the signal and be sent, right, usually down some sort of phosphorylation cascade and then some sort of response. We call this a growth factor. And a lot of times the response is basically a molecule will get into the nucleus of the cell, and it will bind to the DNA, and it will turn on a gene or off a gene. And these genes, when they're turned on or off, can basically create more or less protein. And this protein can act as the signal that tells the cell, hey, it's time to move on into the next step of the cell cycle. We'll look closely at this. Now, what do these molecules look like? Well, we can go back to what we learned last unit. They are kinase enzymes. Remember, kinase enzymes are special enzymes that have the ability to take a phosphate group and phosphorylate another molecule, turning it on, changing its shape and turning it on so that a signal can be passed, right, to the next kinase, and the next kinase, and the next kinase. So in this example, we're going to look at a special type of kinase called cyclin-dependent kinase. What does this mean? Well, it depends on something called a cyclin. So it cannot be activated without another molecule binding to it called a cyclin. Okay, so let's look at here what those mean. So a cyclin, it's a protein. It's a regulatory protein. So its job is to tell the cell when it's time to stop or time to start the next phase of the cell cycle. And here's how it works. It's going to be binding to another molecule called a CDK. The CDK, like I just told you, is a kinase. It's a cyclin-dependent kinase. So it only activates when it's linked up with this special cyclin molecule, this cyclin protein. Some examples, MPF for the G2 checkpoint. So at the G2 checkpoint, if you have this CDK and cyclin complex, that is the green light. This allows the next phase to happen. So we call it the maturation or mitosis promoting factor. It's going to tell the cell at the end of the G2, hey, you can move on into mitosis. Everything's clear. The other one is called anaphase promoting complex. At the M checkpoint, this tells the cell, hey, guess what? All the sister chromatids are connected to kinetochores. You can move on. You can split the sister chromatids and move into anaphase, right? We wouldn't want to leave any chromatids behind. So this anaphase promoting complex needs to be present in order for the cell to move into the next phase, telekinesis. So how does it work? Well, let's say we need a cell or we need a protein to turn on in order to tell the cell that it's the green light, we can move into the next phase. So we have our CDK, our cyclin-dependent kinase, but it's not connected to the cyclin. So our protein is off, right? That's the red light. We can't move into the next phase. But let's say we increase the amount of cyclin in the cell. Then there's so much of the cyclin kind of acting like a substrate to bind to this enzyme, right? And when it binds to the enzyme, because there's such high concentration of the cyclin, it's very saturated, it will bind to the CDK enzyme. And notice what happened here. The CDK, the kinase, will be able to take its own phosphate and phosphorylate our protein. Now, what happens when you phosphorylate a protein? It changes its conformation, it activates it, and turns the protein on. All right, so how does this connect to the cell cycle? Well, let's take a look at our G2 checkpoint here. Okay, so here's our G1, our S, our G2, and our M. Remember, in the G2 phase, what we're doing is we're preparing for mitosis. We're about to head into cell division, but we need to make sure that everything's okay before we move into mitosis. We wouldn't want anything to go wrong before. So we see here that in the G1 phase, we have a bunch of CDK, but we have no cyclin. So the CDK is not binding to the cyclin. It's not turning on any enzymes. But there's going to be a checkpoint here. But once we get down here to the G2, the CDK right at the G2 checkpoint will bind to cyclin. Okay, and once it binds to cyclin, now you have your MPF, your mitosis promoting factor. So it's a complex with the CDK and the cyclin bound. So now we can activate the right enzyme that's going to allow the cell to move into the M phase, mitosis. Once it gets into mitosis, we degrade the cyclin and the cycle can continue on. All right, now during different stages of the cell cycle, you're going to have more MPF and less MPF, right? This mitosis promoting factor, this complex here. And notice that, let's say you're in the M phase, right? So this is showing M, G1, S2, and the next cycle, M, G1, S2, and the next cycle. We're looking here at this graph at the relative concentration. So how many of these molecules do we have? And notice that when the cell is in the M phase, we have a lot of MPF. MPF activity is high. So the MPF is working, it's turned on basically. And notice here that we have a lot of cyclin too. Why do we have a lot of cyclin? Well, it's because the cyclin has now increased its concentration so it can bind and it can be attached to the CPK, which can turn on the MPF, right? And then it will go away. So it'll be degraded. And so the MPF turns off, right? It's not working because it doesn't have a cyclin attached. But then once you get to the next M phase, the cyclin concentration will increase, which will increase the M phase as well. Now, where do these cyclins come from, right? Once they got degraded, how do they get put back together? Well, that's actually run by your DNA. Your DNA has the instructions for building more cyclins. And so when it's time to get to the G2 checkpoint, certain genes in your DNA will be turned on, and these genes will basically create the cyclin protein. You have genes for these proteins, and then once they get made, now you can actually turn on your MPF, which allows your cell to go into the next phase. All right. Now, at the end, though, the end of a cell's life, it's time to stop the cell cycle. We don't need to go on forever. And the type of, this is called programmed cell death, right? It's a type of programmed cell death program because it's all scheduled. Like, this is not a surprise to the cell. So we call it apoptosis. And this occurs in cells sometimes that are infected or damaged. So if the cell has mutated DNA, that DNA could basically create too many cyclins, and those cyclins can create too many MPFs and tell the cell, hey, it's time to keep dividing, even though you don't want to keep dividing because your cell has mutated DNA. That would be a big problem to the daughter cells. You don't want to give your daughter cells mutated DNA. So the cell needs to kill itself, right? And so that's why a cell will go through apoptosis to stop proliferate or stop making copies of a cell that's been damaged. Or to end their life. It's time for the cell to die. There doesn't need to be more cells. Maybe there's too many cells, the cell needs to die. So here's a normal cell, and then here's a cell going through apoptosis. Basically, it's bursting, it's going to burst. All right, so what happens if a cell, though, does not do apoptosis? So here we see a normal cell, and the normal cell will divide, right? It'll go through its M phase, and then that daughter cell will go through an M phase, right? The whole cell cycle, and eventually it divides. But let's say this cell is damaged, right? But there's no repair. The cell will say, okay, you know what, we'll take the hit, we'll sacrifice ourselves, cell suicide or apoptosis. So then we're clear, right? The cell's been damaged, but at least we don't make any more copies of that cell. But with cancer cells, what happens is you have a cell that has been mutated. So when that cell has been mutated, it can make copies of itself with that same mutation. Now, that's not always a big problem, but let's say that cell has made a copy here, right? And here's some more copies. And that cell, again, has another mutation in the DNA. Mutation is basically a change in the DNA. Now you have a third mutation, and we're still okay. But after the fourth mutation, that there's too many mutations in the DNA. And what's going to happen is you're going to have uncontrolled cell growth. So the cell is going to keep going through mitosis without any control of the cell cycle. So the cell, basically all the checkpoints are going to go away. And the cell cycle will keep going and going and going without any regulation, without any stopping. And we saw that when we looked back here at the example, right? When cells are on this in the single layer, they keep dividing even though they know they should stop dividing once there's a single layer. They divide without control, even though they're bumping up against other cells and they're communicating, hey, there's too much. We don't have enough space. There's not enough nutrients. There's not enough food. Still, they're continuing to grow. And that's because the cell signals that tell the cell, hey, it's time to stop cell division, are not working properly. What happens if a cell has uncontrolled cell growth? What happens if a cell has lost control of the cell cycle? It's unable to regulate the cell cycle. Well, again, it will continue to divide and divide and divide. It will continue to go through the cell cycle without regulation, without stopping, and it will form a tumor. So here we have epithelial cells. These are skin cells, and we see a tumor is forming. And right now, the tumor isn't really affecting too many cells around it. But eventually, if it gets too big, you'll start to notice it right here. And the problem becomes when it starts to get into these muscle cells here, which can cause pain. But then also, if the cancer cells get into the bloodstream, now they can move on to other parts of the body because your bloodstream is kind of like a highway. Your blood vessels carry these cancer cells are carried to other parts of your cell. And when the cancer begins to spread, when these tumors or when these cancer cells begin to spread, we call this metastasis. The signal or the cells are spreading to other parts of the body. Here's another Here's another picture. Here is some cell tissue, and we have the primary site of the tumor formation. And once it gets into the bloodstream, then it will be spread out, it will be disseminated or spread to another piece of the cell or the body. And then it will now will leave the bloodstream and go into new tissue and start to increase. And in this case, we're looking at the bloodstream is leading up to the brain and metastasizing and moving and spreading into the brain here. All right, so why are cancer cells created? What do they look like? Well, they're abnormal cells. They're not functioning properly. They're defying the normal regulation of the cell cycle, right? So normally your cell cycle is in the G1 phase, then the G2 phase, the S phase is making a copy, then G2 phase, right? And then it's dividing. But if there's no regulation, then this can happen too quickly. Or if the regulation is not, maybe if there's some damage in the cell can still divide without repairing that damage. And the problem is these cancer cells can invade and colonize other areas and spread and create larger tumors. So carcinogenesis, genesis means new, carcino means cancer, is a development of cancer, but it's gradual. It could take decades. So if you have a normal cell, the DNA gets mutated. Now, that's not the huge issue if there's one mutation in your DNA. But again, like I showed you before, if there's a second mutation or a third mutation or a fourth mutation, we call this a malignant cell. It's a bad cell. And this can now have really, really detrimental cell growth that leads to a tumor. So what's the difference between cancer versus normal cells? Cancer cells, they have abnormal nuclei. Normal cells have normal nuclei. Cancer cells are non-differentiated. Differentiated means you could have bone cells or muscle cells or white blood cells or brain cells or nerve cells, right? So cancer cells, they don't really have an identity. They're non-differentiated, whereas normal cells are differentiated. There's different types in your body. Cancer cells do not undergo apoptosis, so they don't have programmed cell death. They won't die. They're going to continue to divide. But normal cells will undergo apoptosis at a certain point. No contact inhibition. This means if cells are contacting each other and this is density-dependent inhibition, they won't depend on there being too many cells around to stop dividing. But normal cells, if there's too many cells surrounding it, it will inhibit cell growth and it will stop. Disorganized, multi-layered, so they're all layering on top. Usually normal cells form in one organized layer. And cancer cells undergo metastasis and angiogenesis. We'll talk about that later. Okay, so again, here's a picture of a normal cell. We have mostly cytoplasm. We have a single nucleus in each of these three cells, single nucleolus where RNA is made, and fine chromatin. So that chromatin is the DNA plus the protein. Whereas cancer cells have just a little bit of cytoplasm. They can even have multiple nuclei inside, right? Because they're like about to divide and maybe they're making copies of the DNA and making copies of the nuclei. They have multiple enlarged nucleoli and coarse chromatin. Now, what happens? Why are these cells dividing too fast? And the problem is mutations in the DNA, right? Mutations in the DNA. And there are two types that are important. The first are called proto-oncogenes. And you can think of these as genes in powerful positions that go bad. So genes are responsible for building proteins, but if they're messing with the amount of protein that's being made or the type of protein that's being made, then they go bad, kind of like a person in a powerful position that abuses their power. Or a tumor suppressor gene is like an assassin gene that fails to do their job. So tumor suppressors are genes or pieces of DNA that make molecules that are supposed to stop the cell from creating a tumor. They're supposed to stop the cell from dividing too much. But if they don't do their job, then the cell will continue to divide. So assassins are supposed to stop something from happening. They're supposed to kill someone, but they're not. And if they're not, then the people will still live. In the same way, if a tumor suppressor gene isn't working, then it will basically stop the cell from controlling itself and it will continue to divide. So let's look closely at these two types. So again, cancer comes from genes in the DNA, and we'll connect this to the cell cycle as well, and mitosis. So normal genes, proto-oncogenes are normal. Basically, they're genes that tell the cell, hey, it's time to start dividing. They're regulatory genes. They're genes that create proteins that tell the cell it's time to start dividing. But when they mutate, they become something called an oncogene. Now, I know there's a lot of words here. It's a little bit complex, but stay with me here. So normally proto-oncogenes stimulate cell growth, so they tell a cell, okay, move on to the next checkpoint. But if the proto-oncogenes are switched on, or they increase expression, this means they make more protein, then this can cause cancer. An example is RAS, which is a gene. If it's turned on, then it will activate a cyclin production. And we know cyclins, when they bind to CDKs, they say, go, time to move on in the cell cycle. So again, what happens here is if this proto-oncogene is turned on, it turns into an oncogene, and this will create too much cyclin, and that cyclin will keep the cell cycle running. Tumor suppressor genes inhibit cell division normally. These are normal genes in your cell. But if you turn those genes off, then they can cause cancer. And we're going to look at an example of a gene called P53. So I know that there's a lot of words. I remember I've learned this so many times, and every time I learn it, it makes my brain hurt a little bit. So please write this down, pause if you need, and it will help you understand better. So again, a proto-oncogene, if it mutates, it turns into an oncogene. And what happens, it will cause cancer. Why? Well, that gene, that piece of DNA, will code for proteins that will continue the cell cycle. Now, I like to compare proto-oncogenes to when you're driving. Hopefully, well, some of you drive probably. And if you press on the gas, then you go, right? You continue to go. So proto-oncogenes are like genes that tell the cell, hey, go with the cell cycle. But when it mutates, it's like pressing on the gas too fast and you can't take your foot off. And you just go, you keep going and going and going and going and going without stopping. So that's an oncogene. It's causing cancer because the cell is continuing to go through the cell cycle, making more copies without any control. So let's take a look at an example here. So here's a normal nucleus, and we have a chromosome here, one chromatid, and you have a gene. It's called a proto-oncogene. And again, a gene is just a section of DNA right here. Now let's say there's something that is damaging the DNA. So that maybe it's UV light or chemicals. These are called carcinogens. They're things that can mutate your DNA. Then now that proto-oncogene has switched into an oncogene, it's become mutated, and this is cancerous. So how does that affect the cell? Well, let's look here at this model of the cell cycle, and it looks a little different because it's kind of shifted in comparison to our other models. But let's look here at G1. Okay, so we have our normal piece of DNA, and we have our normal proto-oncogene. Oncogene is like the gas for the car. It tells the cell, hey, okay, you can go, continue to divide. But again, let's say that gene is mutated. So this little squiggly line is like UV light or some sort of damaging thing from the environment. Then it will mutate the gene into an oncogene. Now, what happens with that oncogene? Well, what happens is you skip the G2 phase. So you're going to automatically stimulate cell division without any growth factor. So you're not going to move into the G2 phase, which is going to basically have that G2 cell checkpoint, which is going to it should stop the DNA or stop the cell from dividing because there's been damage. But instead, the cell will basically skip the G2 phase and it will go straight into copying cell division, okay, without any regulation, without stopping. And so once that cell divides, now the two copies have a mutated piece of DNA. Now, this isn't totally a problem. Just one mutation isn't a big deal. But again, over a lifetime, if there's multiple, like 4 mutations in the DNA from multiple exposures to light or chemicals, then this could really cause a problem. So here's another example, kind of like an image. So normally you have signals called growth factors. Again, this is a ligand. It's a hormone here. Maybe this came from a pituitary gland in your brain or some other part of your body, and it's connecting to our receptor protein in the cell membrane. Now, this is going to activate a protein called RAS, and that protein now is called a growth factor. And it's going to go into the nucleus and turn on genes and tell the cell, okay, time to create cyclins or proteins that are going to continue the cell cycle at a normal pace. You're stepping on the gas here by creating proteins from the DNA that are continuing the cell cycle. But remember, oncogenes are kind of like powerful people that have abused their power. This RAS protein, if there's been some sort of mutation in the cell, then it actually has the ability to go tell the DNA, hey, time to make cyclins. It will work as a growth factor. It will go into the DNA without any growth factor here, without any ligand here. bonding to activate this. It's always going to be activated. It's going to be consistently activated. And so it will continue telling your DNA, make cyclins, make cyclins, make cyclins. And that cyclin will bond to a CDK. That's the go ahead. That's the green light. And so you're always going to have the green light on without anything stopping. Typically, this growth factor is telling it to turn on, and then the growth factor can go away until everything is shut down, stopping everything, right? But this is like the RAS has gone out of control. It's stepping on the gas without anything telling it to stop. Okay, so we talked about proto-oncogenes. Now we'll talk about tumor suppressor genes. So a tumor suppressor gene is normal. It's good. It stops tumors from growing. It stops a cell from not being able to regulate its cell cycle. But if this gene mutates, it will no longer produce proteins that stop the uncontrolled cell cycle. So tumor suppressor genes, normally they're good. They act like brakes to the cell cycle. They put on the red light saying stop. But if you lose this ability, if you can't step on the brakes, then again, your cell will continue to divide without anything controlling it. So here's a picture. Here's a gene. It's a piece of DNA, and its job is to create a protein. And this protein is growth inhibiting. Will stop the cell from dividing too much, right? So we're only going to divide slowly. Our cell divisions are under control. These cells are not going to divide. These cells are going to divide. We're all good. So again, maybe this piece of DNA creates a protein that will be secreted into the cells around it. And so if it hits this one, okay, tell that cell to divide. But we only have a little bit of it, so we're not going to tell these cells to divide. So we're not creating too many new cells. But let's say you have a mutated tumor suppressor gene. So this piece of the DNA has been changed. Now you're going to make a defective protein. The protein is not going to work properly. It's not going to actively be able to bind to cyclins or stop the cell cycle from continuing. And so the cell cycle will not be under control. All the cells around will continue to divide. Uncontrolled cell growth. So again, this is like brakes failing on the car. So normally you have your chromosomes here, and they are able to divide. But let's say you have one mutation. Okay, so you have a mutation here. You still have the other sister chromatid is working properly here. And so you can actually have normal cell growth. But if you have sister chromatids that have mutated genes on both of the sister chromatids, then you'll have you'll have lost the tumor suppressor gene, and you'll have the brakes are going to fail, and you're going to continue making new copies. This isn't totally important for you to like fully understand, but just kind of like a preview into our DNA unit, next unit. Okay, so again, here's another picture. If a cell receives a growth factor receptor protein, it's going to turn on our G protein here, right? And our G protein is going to be able to transduce a signal. So phosphorylation cascade, a bunch of kinases will phosphate each other until they get into the nucleus, and they work as a transcription factor, turning on a piece of DNA that makes a molecule, right here, this protein that will start the cell cycle. So this could be probably like a cyclin that will bind to a CDK, go, green light. But let's say there's a mutation. You don't even need a ligand. You don't need any growth factor. The RAS protein works on its own, transducing a signal, even though there was nothing telling it to start, right? There's no other hormone in the body that's telling it to start dividing. And so the DNA will have the activator binded forever, and this won't be able to come off. And so you'll create, from your DNA, you'll create so much protein all the time. Overexpression, we call this. So too much of the DNA is being copied and turned into protein. And so now you'll have too much go green light, and you'll have increased cell division. So what is cancer? Essentially, it's a failure of cell division control. The cell cycle is continuing unrestrained, uncontrolled cell growth. So what control is lost? The checkpoints stops are not working properly. So you're not going to be able to say green light or red light. It's just green light all the time. Now, gene P53, so there's a piece of your DNA, everyone has it, and it plays a key role in the G1 checkpoint, so getting from G1 into the S phase. Now, normally, the P53 is a protein in your body, and it's made from the P53 gene. So genes are in the instructions for building proteins, so the P53 gene will create a P53 protein. And its job is to stop cell division if it detects damaged DNA. So it can do a couple things. First, the P53 protein can stimulate repair enzymes to fix your DNA. So if your DNA is mutated, or if it's damaged, let's say from UV light or from a chemical, then it will say, oh, So fix your DNA before we move into the S phase. We don't want to make a copy of the DNA if it's not perfect. Or it can force and keep the cell in the G naught or the resting phase. So basically, if you have a lot of p53 protein, the cell has the red light on, it's still in G naught. We don't want to move into the S cycle or the S phase. Or the p53 gene, let's say we really don't want to go into any more cell division ever again. We can start apoptosis, and we can kill the cell if the cell is too damaged. Now, in most cancers, in order for them to create a tumor or basically stop regulating cell growth, their job is to shut down p53. So that's how a lot of people get cancer, is if their p53 gene isn't working, because again, the p53 gene's job is to fix DNA or to stop the cell cycle or cause apoptosis. So here's how it works. So let's say your DNA is damaged. Well, if your DNA is damaged, then, like for example, heat or radiation or chemicals that could mutate your DNA, your cells are going to make p53 protein. So p53 is a protein that can latch onto your DNA and say, hey, don't go into S phase. We don't want to replicate. We don't want to make a copy of the DNA. And then two options from here. Either there's going to be an enzyme, another protein in your cell that will go and it will fix this DNA piece. And then once it's fixed, then the cell can continue to divide. So the p53 is there. It will say, hey, okay, keep dividing. The DNA has been fixed. Or let's say you have the protein is attached to the DNA. Let's say it's too bad. We can't fix the DNA, right? It's too broken. Then it will trigger enzymes to repair the damaged region. But if it's too beyond repair, it can't really be fixed, then the p53 will trigger apoptosis and it will destroy the cell. So this is good, right? We don't want damaged cells, and it helps to fix the cells, right? But let's say we don't have p53, it's not working properly. So if our DNA is damaged, we don't have working p53, nothing can bind to the DNA to say, hey, stop dividing. And so this piece of DNA will move into S phase and it will make a copy of itself, a sister chromatid. And when that happens, there's nothing to repair the DNA, and there's nothing to tell the cell to stop. And so the cell can normally it's supposed to move into normal cell division. But if the cell is damaged too much and the DNA is damaged too much, then it will keep dividing and dividing and dividing. And if there's more mutations, eventually, if there's more mutations here, it can turn into a cancerous cell. So not all of the cells will be cancer cells, only if there are more mutations later on. All right, so just kind of a summary. Typically, cancer develops if one cell line experiences about 6 mutations. So here we're looking at just one mutation, but again, if the cells over time experience about 4 to 6 mutations, then it becomes a really damaged cell. So some things that can happen. You can have unlimited growth. So if you turn on oncogenes are like the gas is going too much, or you can ignore checkpoints. So you turn off the tumor suppressor genes, you turn off the brakes, and the cell will continue dividing without any brakes. You can escape apoptosis, so you can turn off the genes that tell the cell, hey, it's time to die. So then the cell will never think it's time to die. Immortality, so the cells will turn on genes that basically allow the cell to continue to dividing without stopping. We haven't quite talked about this, but just another example. Promotes blood vessel growth. So again, these are other examples, not super important. But we did learn about this, overcome anchor and density dependent. So we're turning off a touch sensor gene. So there are certain proteins in the cell membrane that allow the cell to recognize cells around it. And so if those proteins aren't created, then the cells aren't able to understand that, wait, there are too many cells around me, we're too dense, we should stop dividing. So they're going to basically keep dividing without stopping. Okay, so what are some things that causes these hits? We call these carcinogens. And some examples: UV radiation, chemical exposure, radiation exposure, heat, cigarette smoke, pollution, age, genetics. But it's not that these things necessarily, like if you're exposed to ultraviolet light, which by the way, you're exposed every day. Ultraviolet light comes from the sun. If you're exposed once to the sun, you're not going to get cancer, right? It would have to mutate your DNA 4 to 6 times in order for that cell to actually turn into a cancerous cell. And it might not even spread into your bloodstream. So if it only stays in one place and it doesn't metastasize, we call it a benign tumor, and it won't spread to the rest of your body. So here's another example. Here's breast cancer. So a tumor will start growing from a single cancer cell. One cell turns cancerous. It will start dividing. It will invade neighboring tissue, and then eventually it will spread through your lymph vessels and your blood vessels. So these are two highways in your body that are going to send the cancer cells to other places until they get to another part of the body and they metastasize. All right, so again, if you have a tumor, a mass of cells, it can be called a benign tumor. It's not totally safe because it can still grow, but they usually stay at the same space. They don't really move until p53 halts cell division. So they don't keep growing because eventually p53 will work and then it will stop the cell from dividing. The tissue that has been invaded by the benign tumor still has the same properties, so it's not going to become damaged too much. The cells around the tumor, but they don't cause serious problems. They can be removed by surgery, not a problem. But malignant tumors, they can leave their original site. They move somewhere else. They lose attachments to the nearby cells, so they don't just stay in one place. They start moving around. They get carried to other tissues, start more tumors. So the movement of cancer cells to another place where another tumor starts is called metastasis. This tissue around the tumor doesn't resemble the original tissue. So the malignant tumors will take nutrients, water, food from the neighboring tissue, the normal cells around, and basically steal nutrients from them and impair functions of organs. So the organ might shut down if too many cancer cells are spreading and invading and taking up so much space. And so organs will stop working, they'll shut down. All right, so there are treatments for cancer, and what these treatments do is they target these rapidly dividing cells. So chemotherapy and high energy radiation with toxic drugs, these medications or these treatments, their job is to try to kill these rapidly dividing cells, but it comes at the expense of healthy cells. So here, hair follicle cells are now going to be destroyed with this chemotherapy because as you're targeting the cancer cells, you're also targeting healthy cells as well. So hopefully from this video, you understand that cells communicate with each other. And we saw an example of this first enduring understanding when we looked at density-dependent inhibition. So cells communicate with each other. They send signals, hey, when is it time to divide or not? And they'll respond to these signals by turning on genes or off genes that tell the cell to create more proteins that will start or stop the cell cycle. The second is there's a timing and a coordination of things that help the cell know when it's time to grow for this specific piece here. And that is dependent on environmental cues. So things outside the cell, like growth factors that are ligands that bind and tell the cell, hey, it's time to start dividing or stop dividing. And lastly, heritable information provides for continuity of life. So we learned about mitosis, how cells make copies of their DNA and divide and put matching copies of the DNA in each daughter cell. So that's the end of this video, and we'll see you in class.